Now let's start with Pachytene stage. That is the next stage of prophase 1. During this stage, chromosomes show further condensation. Chromosomes exhibit further condensation, and hence these chromosomes appear very thick and short. Chromosomes exhibit further condensation, and hence these chromosomes appear thick and short. Thick and short. Okay, so during this stage, each chromosome is with 2 chromatids and it appears to be having 2 chromatids. So 2 chromatids are visible in each chromosome. 2 chromatids are visible in each chromosome. 2 chromatids are visible in each chromosome. Previously, during this leptotene and zygotene, each chromosome is with 2 chromatids, but it appears to be having only 1 chromatid. But during this stage, in each chromosome, 2 chromatids are visible. Now let's see those chromosomes. Homologous pair of chromosomes. Okay, this is a homologous pair of chromosomes which have been paired during zygotene stage. Okay, now this is one bivalent or tetrad. So, here these 2 chromatids of a chromosome are called sister chromatids. These 2 chromatids are sister chromatids. And these 2 chromatids of a chromosome are also called as sister chromatids. But the chromatid of this chromosome becomes non-sister to the chromatid of this chromosome. So, these 2 chromatids are called as non-sister chromatids. These are non-sister chromatids. Okay, so during this phase, there is appearance of recombination nodules. During this phase, that is during Pachytene stage, there is appearance of recombination nodules. So these recombination nodules are the sites of crossing over. These are the sites or the areas where crossing over occurs. These are the sites or areas where crossing over occurs. So now let's see what is this crossing over. So crossing over is exchange of genetic material between the non-sister chromatids of homologous chromosomes. That is called crossing over. So exchange of genetic material between non-sister chromatids of homologous chromosomes. So these 2 chromatids are non-sisters, but they are homologous to each other. These chromosomes are homologous to each other. So here what is happening? So a part of this chromatid is cut and it is joined with its non-sister chromatid. And a part of its chromatid is cut. Chromatid is made up of what? DNA. DNA is nothing but genetic material. So a part of this its chromatid is cut. This chromatid is cut and it is joined with, reunited with this chromatid. So what is taking place? Exchange of genetic material is taking place between the non-sister chromatids of homologous chromosomes. So after exchange of genetic material, they appear like this. After crossing over, after exchange of genetic material, they appear like this. Its segment is shifted onto this chromatid and its segment is shifted onto this chromatid. So this is called crossing over, which is the exchange of genetic material between non-sister chromatids of homologous chromosomes. So crossing over is exchange of genetic material between non-sister chromatids of homologous chromosomes. Non-sister chromatids of homologous chromosomes. Homologous chromosomes present in a bivalent. Chromosomes, right. So this crossing over is performed by an enzyme called recombinase. This crossing over is performed by an enzyme called recombinase, and this process is also called as recombination. Let's see why it is called as recombination. So due to the exchange of genetic material between non-sister chromatids of homologous chromosomes, there is exchange of genes. There is new combination of genes, that is recombination of genes. Due to new combination of genes or recombination of genes, it results in the formation of new combination of traits or recombination of traits. Hence, this process of crossing over is also called as recombination. And this crossing over is performed by an enzyme, by the activity of an enzyme, recombinase. So this recombinase enzyme is a multi-enzyme complex. This recombinase enzyme is multi-enzyme complex. This is a multi-enzyme complex because it consists of two enzymes. Those two enzymes are endonuclease and DNA ligase. So those two enzymes are endonuclease and DNA ligase. Let's see the functions of these two enzymes present in this multi-enzyme complex called recombinase. Okay, endonuclease is involved in cutting the genetic material from one chromatid. DNA ligase is involved in ligating or joining this cut part onto its non-sister chromatid of its homologous chromosome. In the same way, this chromatid, some part of this chromatid is also cut by the activity of endonuclease, and it is reunited with its non-sister chromatid of homologous chromosome. So, this enzyme recombinase is involved in performing crossing over. So, on these chromosomes, crossing over is taking place. At the site of these crossing overs, at the sites of these crossing overs, these chromosomes appear dense or dark. Okay, so these dense or dark colored appearance, dark colored points are called as recombination nodules, because at these points, recombinase enzyme is working. Hence, these points which are appearing dark or dense at the point of crossing over are called as recombination nodules. So what can we say? Pachytene is marked for what? Pachytene is marked for crossing over or recombination, which is performed by the activity of an enzyme recombinase. Okay. So these are all the changes that occur inside the nucleus of the cell during this Pachytene stage of prophase-I of meiosis. Now, what are the changes that occur inside the cytoplasm of the cell during this Pachytene stage? So the changes that occur inside the cytoplasm is the centrosomes or the asters, which have already started moving from one another, moving away from one another. They move further away from one another, which try to reach the opposite poles of the cell, which represents the pole of spindle apparatus. So this is about Pachytene stage. In the next class, I will be starting with diplotin and then diakinesis. Thank you, students.